everyone. This is Ron Hayes. I'm the producer for this session. I'd like to introduce you all to Khaled Rajub, who will be leading the session, leveraging knowledge graph and imagining to predict gene disease links in cardiovascular diseases. Rajub, the floor is yours. Take it away.
Can you see my slides? Sorry, let me check the chat. Yes, okay, perfect. So, hi everyone, I'm Khaled. I'm a postdoctoral researcher in data science at MRC Laboratory of Medical Science at Imperial College London. And today I will present my work on using knowledge graph and imaging data to predict novel gene disease association in cardiovascular diseases. So I work in the computational cardiac imaging group led by Professor Declan O'Regan at Imperial College London. And in my talk, I will go through this agenda. So a little bit background about predicting gene disease association. So genome-wide association studies are the primary tools that have been used to find the association between the gene variants and the disease. However, those tools are limited to a single data modality, so they cannot include like different biological data modalities. So our approach is to construct a multimodal knowledge graph that integrates different databases, biomedical databases, and to predict gene disease associations specifically for cardiovascular diseases, including heart failure, atrial fibrillation, and myocardial infarction. And also we assess the impact of incorporating imaging data, cardiac imaging data, on the performance of the knowledge graph. So we used Neo4j and Python as tools to run our analysis. So we extract the data from the UK Biobank data mainly, and from other 18 external biomedical databases. And from the UK Biobank data, we used the cardiac MRI, which is called cardiac magnetic resonance, and the short name is CMR. So from those images, we extracted features like the structural features for our heart using convolutional neural network. And those features are called CMR features. So I will be using the term CMR features very frequently in my presentation, which refers to the extracted features from the cardiac MRI images. So those extracted CMR features are associated with certain anatomical regions in our heart. So for example, the right ventricle ejection fraction, which is RVF, is linked to the left ventricle, the right ventricle, and the left ventricle ejection fraction is linked to our left ventricle. So we use this schematic graph in this slide to build our knowledge graph, and it shows how we integrated the extracted CMR features in the knowledge graph. So the CMR features are integrated in our knowledge graph as nodes, and the value of those CMR features are integrated as properties on the link between the patient or the individual and the CMR feature. And then we ran the cipher code in this slide to create the nodes in our knowledge graph. And I just want to highlight that in our patient's node, we integrated the demographics of the patients inside the patient's node. So each patient's node has the demographics of the patients, including the body mass index, the BMI, the body surface area, PSA, ethnicity, sex, and age at MRI. And also on the relationship between the patient and the CMR features, we have properties, and that those properties have the value of the CMR features that are linked to the patients. And to do downstream tasks, such as predicting new links in the knowledge graph, we converted the knowledge graph from the high dimensional representation into a low dimensional representation using embedding algorithm. So we didn't use the traditional embedding algorithms such as Node-to-Vic or like Trans-E, because those embedding algorithms do not take into account the properties on the links and the properties inside the node. So we used our own embedding algorithm, which is called directed variational graph auto-encoder, which takes into account the direction of the relationship between the nodes, the features inside the nodes, the features on the links between the nodes, and the node type. After generating the embeddings from the knowledge graph using our embedding algorithm, we extracted the embeddings for the gene and the disease of interest. So from the embedding layer, we extracted those embeddings, and then we classified them into positive genes and negative genes. So positive genes mean, in our case, genes that have known association with the disease of interest. Negative genes mean genes that have no association with the disease. And we use the database called disease gene network as a ground truth to label those positive genes and negative genes. And then we concatenated those positive genes and negative genes with the embedding of the disease of interest. After concatenating the embeddings, we applied 3 traditional machine learning classifiers, including random forest, support vector machine, and artificial neural network. And then from those classifiers, actually, we selected the classifier that outperforms the other classifiers. So, for example, we picked support vector machine, and then we used the selected classifier to predict unknown association between a new gene and disease. So we predicted those associated genes, and then we considered the top 10 predicted genes based on their generated probability to do the enrichment analysis, or to validate those predicted genes to see if they're really associated with the disease or not. So we took those 10 top predicted genes and did the enrichment analysis for those genes to see if they are significantly associated with the critical pathways for the disease of interest. And after that, we evaluated the importance level of the CMR features that we integrated in the knowledge graph and the importance for the other nodes in our knowledge graph using the PageRank score algorithm. And also we did an application study to evaluate the importance of the CMR features. So we created 2 versions of knowledge graph. One knowledge graph that has the CMR features integrated. The other knowledge graph doesn't have the CMR features integrated. And then we ran the same analysis, which is predicting the gene disease association using both different knowledge graphs. And then we evaluate the difference between the two knowledge graphs based on the predicted genes and based on a number of the enriched pathways. So from the 19 databases that we used, we extracted 12 entities. And in the individual entities, which refers to the patient entities, we have around 7,000 patients, actually 7,000 individuals. More than 90% of them comes from different cardiovascular diseases, such as heart failure, myocardial infarction, dilated cardiomyopathy. And a part of them are normal individuals, so they don't have any cardiovascular diseases. And in total, we ended up with 33,000 nodes in our knowledge graph and around 1,100,000 relationships between those nodes. And I just visualized like a subgraph from the whole knowledge graph that we have. And then using our embedding algorithm, we obtained a high accuracy, which is equal to 98%. And this accuracy was calculated by comparing the original graph with the reconstructed graph. And after extracting the embeddings from the knowledge graph, we started with the first downstream task, which is using a supervised machine learning algorithm to classify the positive genes and the negative genes for the disease of interest. And we found out that support vector machine obtained the best performance in terms of predicting the positive genes and the negative genes for the 3 cardiovascular diseases: heart failure, atrial fibrillation, and myocardial infarction. And since the support vector machine obtained the highest performance, we selected that model to predict the unknown association between the gene and the disease for the 3 cardiovascular diseases. And we used the top 10 predicted genes for each disease to do the enrichment analysis. And I will start with the heart failure. So that out of the top 10 predicted genes in heart failure, actually around 8 genes were critically or were significantly associated with 66 critical pathways associated with heart failure, which emphasizes that those genes are more likely associated with heart failure. In the second enrichment analysis, we selected the genes that are predicted for atrial fibrillation. And out of the top 10 predicted genes, 7 genes were significantly associated with 14 critical pathways for atrial fibrillation. And in the third enrichment analysis, we found out that top 10 predicted genes for myocardial infarction were significantly associated with 42 critical pathways for myocardial infarction. And as I mentioned before, we use the PageRank score to evaluate the importance of the CMR features and the other entities in our knowledge graph. So as we can see in this slide, the CMR features obtained the highest score compared to the other entities in our knowledge graph, followed by the exposure, gene, and disease. And among those CMR features, we found out that the left ventricle ejection fraction obtained the highest score among the CMR features. However, the difference between those CMR features regarding to the PageRank score was very marginal. So the PageRank score here tells us the influential level for each CMR feature node in our knowledge graph. And in the ablation study that we did to find the contribution of the CMR nodes in our knowledge graph, so the figure on the right-hand side represent the performance using the knowledge graph without the CMR features, and the figure on the left-hand side represent the performance using the knowledge graph with the CMR features. And we can see that including the CMR features in the knowledge graph enable the knowledge graph to predict genes that are associated with more critical pathways for the disease of interest. While when we don't include the CMR features in the knowledge graph, the number of critical pathways for the disease of interest are very small compared to the one with the CMR features, which emphasizes that the CMR features that CMR nodes actually are very influential in our knowledge graph. So just to sum up, so the knowledge graph was very helpful in our work in terms of integrating multimodal data. So it enables us to study human diseases such as cardiovascular diseases. And our knowledge graph provides a holistic view of cardiovascular diseases by integrating data from 19 different biological databases. And this approach uncovers novel candidate genes and potential therapeutics for major global causes of morbidity and mortality. And it demonstrates that disease-specific semantic models can be significantly enhanced using cardiovascular imaging data. Thank you very much, and I'm happy to take the questions. I'm just trying to answer the first question. Hi, please, do you have a research? Yes. So actually, we have a preprint on this work, and it's going to be published maybe in the next few weeks, actually, on Nature Cardiovascular. So yeah, the next questions. You say novel candidate genes, but they are all part of known pathways. Do you mean novel? Yes, I mean novel here in terms of classifying them or adding them in the standard databases as they are associated with the disease of interest. So those genes are not mentioned in those ground truth databases to be associated with cardiovascular diseases, but we found them based on the enrichment analysis associated with the critical pathways but those critical pathways are associated with cardiovascular diseases. Yeah. Yes, they are, but those genes still are not listed as positive genes for the disease of interest. So yeah, for integrating this, so actually we extracted the data from each database separately. So we used our Python script to extract the data, and then we saved them in like a CSV files, and then we uploaded those CSV files into Neo4j, and then we built the knowledge graph using Neo4j, and then we used Jupyter Notebook to retrieve the knowledge graph from Neo4j into Jupyter Notebook environment. And then we were able to do the embeddings, the embedding algorithm, and to perform the downstream task, which is predicting the links between the gene and the disease. So I will just type my email in case anyone wants to ask more questions or to reach out. Thank you very much.